Jeanne Marrazzo. I met her last year at the NIH, and she's such a wonderful and vibrant and positive person. I think you will all enjoy talking to her, and I'm so thankful, Jeanne, for that you're coming to the faculty and doing a talk for us. Um, she took over the directorship of the NIH about a year ago now, and is moving forward with a lot of projects there. Her background is in infectious disease. She has done a lot of work on infections, allergies, immunological things, and is also recognized for her groundbreaking work on sexually transmitted infections, with a particular focus on women's health. Before she joined the NIH, Dr. Marrazzo was the director of the Division of Infectious Disease at the University of Alabama in Birmingham. And there she led significant research efforts and championed a lot of development and training, especially for underrepresented and minority groups in medicine. She's well authored. She's got more than 250 publications. She's a fellow of the American College of Physicians and of the Infectious Disease Society of America. She has mentored and trained so many people, and it's a real privilege to have you here, Jeanne. Thank you for visiting Stellenbosch University, and we hope you enjoy your time in South Africa.
Can you guys hear me and the mic? Okay, great. Well, it's an incredible pleasure to be here. I have admired the work that people have done here for many, many years. So I want to thank Elmi for that very kind introduction and for all of you for hosting all of us today. And I have to take full responsibility for the slide situation. I forgot my thumb drive, the USB drive that had the slides on it. So it's all on me. I thank you for your patience, and I thank the team for being patient with me. So what I'm going to do today, to talk about science to this group is very daunting because you have some of the world's most knowledgeable, most accomplished scientists and clinical trialists in the world, right? So I'm not going to give you a big new scientific overview. What I'm going to do mostly is to talk about why our relationship is so important. Why NIAID needs to have a very vibrant relationship with you in order to really accomplish our mission, which is shown here, to support and conduct biomedical research to better understand and treat infectious, immunologic, and allergic diseases. That building on the left there is our Vaccine Research Center, which many of you know, and we've worked very closely with many of your teams. On top is our Rocky Mountain Lab, which is in Montana, where we have our biocontainment facilities as well as a lot of animal facilities. And then below is our Fisher Lane place in Maryland. So I want to just start by recognizing the extraordinary history that we have together. We have over 35 years of collaboration. And I think it's one of the oldest established research relationships that NIAID has with any other country. We also have, we also have established in 2013 a bilateral agreement, which is an MOU between us and the South Africa Medical Research Council. And that's summarized below. We've been awarding those grants in now what's coming up on the third cycle. So we're really excited about that. But to give you a sense of how much we fund and what we do here, let me just make sure. South Africa is actually the largest recipient of NIAID international funding, and that amounts to about $90 million a year. So it's a very extensive investment. And it's wonderful to come and see the physical and human yield of that investment here today. So a couple of the things that we have done, I want to emphasize, we are very much known for our basic science, but we are also known for basic science as it translates to impact and to much of the work that you're doing here. And this slide just gives you a couple of examples of things that grew out of discovery, largely in the laboratory, but then became, I would say, networked and disseminated in ways that have really impacted public health. One of my favorite examples is, of course, Barney Graham's leadership of the understanding of the respiratory syncytial virus F protein, how you lock it in the pre-fusion state before it binds to the cell membrane so that you can then generate an appropriate and protective antigenic response. Of course, that work informed the ability to do the same thing with SARS-CoV-2. And that's why, or one of the reasons why we were so able to develop those vaccines so quickly. So really important. And I should just mention that this is built on many years of investment in the HIV vaccine field. So much of the structural biology that has allowed us to characterize these viruses is because we have spent decades investing in trying to get an HIV vaccine. And I'll come back to that in a little bit. Um, and the other one I'll mention is just the HPTN, and I'm going to talk a little bit about the HPTN. I know this talk is not very heavy on HIV. Again, you are the epicenter of not just HIV and the disease, but also research. So we'll come back to that in a minute. So what do we do and what are we focused on right now? So we have a very strong interest, of course, in emergent or resurging infectious diseases. And if you take a look at what we're dealing with right now, a lot of it is related to zoonotic spillover. And of course, we estimate that about 3/4 of our emerging pathogens are actually zoonotic. If you look at outbreaks in the United States and multiple other countries, you can see those are the infections that we're focusing on there. And then outside the United States, a number of infections listed in that right-hand slide. I think one of great interest recently is Oropouche virus. If you haven't heard about that, that seems to be really emerging strongly in Latin America and may unfortunately have some perinatal effects that are similar to or reminiscent of West Nile virus. So concern for fetal developmental delays. So we're really interested in that, working with our colleagues in Latin America to do that. Of course, our major area of work with you all has been largely in TB and HIV, not exclusively. And HIV has been a gigantic part of our portfolio. And of course, Dr. Fauci was a champion of this and is well known for his work in HIV. We have about 1/3 of our budget actually allocated to HIV. So it's a substantial investment. Some of that, much of that is actually congressionally mandated. So we have a governmental congressional mandate to do this work on HIV. Bless you. So what do we do for HIV related research? It really intersects with our other areas of interest because in our non-HIV infectious diseases, that's where tuberculosis falls, that's where hepatitis B and C fall, and that's where many of the other opportunistic infections that affect people living with HIV fall as well. And of course, understanding the basic immunology of HIV is a major issue. So another big bucket of what we do is pandemic preparedness. And I think many of you are well aware of our role in this. And I understand that you all had many calls with our leadership to inform the trajectory of research and especially defining the emergence of Omicron and other variants. So one thing that is really quite interesting when you think about all the pathogens that we are technically responsible for, it's something like 320. So there's no way, even when you break down those that are likely to be of concern for a pandemic, you simply can't prepare for every single thing that might emerge, especially when you have things emerging that you have never seen before, like SARS-CoV-2, for example. So we have a pandemic preparedness plan that's shown there that takes this approach of a prototype and priority pathogen approach. And what we're doing is essentially developing strategies that focus on viral families, some bacteria, but mostly viral families to make sure that, for example, if an Oropouche virus comes out, we have a prototype antigen, a prototype immunogen that we can actually start slotting into our platforms to take a look at it. So the idea here is to be prepared for pretty much all of the major families that could come out in a pandemic. Couple that with surveillance, which I know you're very interested in, and I'm eager to hear more about, to identify novel threats before they emerge. And we've got some really interesting programs, including Danny Duick's Rat Pig program. If you haven't heard about that, we can talk about that. And then make sure that we have the infrastructure and technology to bridge gaps in research and do the preclinical and clinical testing. And again, it's one of the wonderful things about being here in person, because I can actually see how well positioned all of you are. And that's what's really needed if you're talking about shortening the timeline between detection of an outbreak and authorization and implementation of countermeasures. So I'll just mention a few of the programs that support preparedness. These are programs that I think are really worth taking a look at. If you're interested specifically in what we're doing, we've got something called REVAMP, which is research and development of vaccine and monoclonal antibodies for pandemic preparedness. These are a network of basic and cooperative research centers with attention to those specific virus families, and different research centers have a focus on different virus families. Again, not everybody can do every single virus, even if they've got the biggest lab in the world. So it's really nice because we've selected these centers so that they can complement each other. Danny Dueck's program is called PREMISE, and that's the Pandemic Response Repository through Microbial and Immune Surveillance and Epidemiology. And what he is doing is a fascinating global attempt to create a pathogen agnostic anticipatory reagent and countermeasure discovery program. So he's collecting samples, sera, lots of things, and really trying to look deeply and interrogate the sera, for example, B cells, for example, what's in there? What are people reacting to? What are we seeing? What are the virologic footprints or fingerprints that we need to be aware of depending on where they're coming from? And then our antiviral program for pandemics is trying to develop a pipeline of antivirals and build sustainable platforms. You know, if you are an infectious disease person, viruses are really hard to treat. We have very few viruses that we have great treatments for. HIV is really an exception. Herpes, we can do some. We have hepatitis C, which is fantastic. But, you know, even CMV, EBV, certainly the respiratory viruses, oseltamivir, Tamiflu, even the new flu agents, they don't work that well. And honestly, for me to think about, as I'll talk about soon, H5N1, relying on oseltamivir for the stockpile for H5N1 makes me very anxious. I stay awake at night thinking about that. So we'll talk about that in a minute. I want to mention too, one thing that I don't think we have done enough on at NIAID that I'm very concerned about is global antimicrobial resistance. And I was talking about this morning at the Desmond Tutu Center because I include mycobacterial resistance in this threat. And I think it's very important that we recognize this. A couple of statistics that I have recently read about with some nice special reports. When you look at deaths from antimicrobial resistance as recently as 2019, the number is about the same as HIV and malaria combined. And these went up quite dramatically during the pandemic. Lots of reasons for this, lots of antibiotic overuse, lots of people in hospitals getting infected with colonized agents that were already in the hospitals. And these are really hard to study because they don't occur in predictable settings. You know they may be in an ICU, but you never know when an outbreak, say, of multi-drug resistant Acinetobacter is going to occur, or a bad Klebsiella. So they can be very hard. Diagnostics are really not available quickly. And the biggest problem, I think, is there's a limited market for new antibiotics, right? Why would pharmaceutical companies want to create really fancy, expensive antibiotics when you're going to use a couple of doses a month, maybe in an ICU, not even globally, right? When you think about what capacity is globally. So it is a really big challenge. I know that Dr. Mueller in particular is an expert in this area, and I know that some of the most pioneering work has been done in Cape Town in transplant, particularly people living with HIV. And I just want to point out, again, as we are more successful with solid organ transplants and stem cell transplants, we are seeing more of these infections in these patients. I think some of the most heartbreaking experiences I have had is to see people who have waited for an organ, they get transplanted, they do great, and then they come in the ICU and then they get a multi-drug resistant infection and they die. It is really awful. It's sort of like, how can we have gotten this far and we don't have ways to treat this? And this is just, I thought, a nice paper that looked at multi-drug resistant infections after liver transplantation. And they are common and they are probably getting more resistant. So as we think about trying to scale up these programs to fix big problems like organ need, we also have to think about the ancillary measures that are going to support the health of people after they have gotten their transplant. So just a couple of programs. I won't go through them all, but we have a number of antimicrobial resistance programs and initiatives that I think people often don't know about because they don't realize that we are involved in this area. Again, I think we need to do more. We also have a congressional mandate to study this because people realize that this is a big threat. The other thing I think that's really important is to think about where these infections are coming from. Many of them are arising from Southeast Asia, places where you can get all kinds of antibiotics over the counter. Started with fluoroquinolones, right? And now has just gone on from there. So if you look at the epicenter, it does tend to be in Southeast Asia. So we really have to think about this, I think, again, as a global problem. There are some interesting things that we're doing that are not reliant on pharma or drug development, thinking in the traditional sense. Some of you may have heard about phage therapy, and maybe you've even read The Perfect Predator, which is Stephanie Strathd's book about her husband's ordeal with multi-drug resistant Acinetobacter. Stephanie's an HIV specialist, PhD in San Diego, and she managed to actually get phage for her husband's life-threatening infection, which he acquired, I believe, when they were abroad in India or after, I can't remember. But that's not always the case. Most people don't have access. The other problem is it's a very bespoke therapy right now. You need to have the perfect phage for your particular bacteria. So if you talk to people who do phage work, they're out there collecting phages from like sewers and all kinds of interesting places. There's billions of phages in the environment. There probably is a phage for every single bacteria that is going to give us a problem, but the problem is matching it. So you can do it a little bit more systematically when you develop these phages for specific infections. And this has been done very nicely, not just for Mycobacterium, non-tuberculous Mycobacterium. You may have seen this for pulmonary MAI, but also for people with cystic fibrosis who were chronically colonized with Pseudomonas aeruginosa. And I think that this is going to be a very exciting area in probably the next decade. And there's a number of phage centers that are really gearing up, again, to do a broader library characterization of potential phages, and then really looking at how we can match those with the areas of greatest needs. The other approach, of course, is to say, okay, these are not diseases that are going to make people a lot of money in our capitalist society. So how can we figure out alternative ways to get new antibiotics out there? Gonorrhea, not exactly a bonanza for drug companies to make a profit on, right? And yet we are seeing the emergence of really significant resistant gonorrhea, and I may have a slide on that in a second. But this is an example of oh, actually, I did include it. There it is. It's that little box on the right. Emergence of extensively drug-resistant Neisseria gonorrhea from France. This was just reported in September. Actually, it's next month, I guess. That's a little strange, but it is it's the EID issue, emerging infectious disease issue for next month. So this is very fresh. These are French cases of two people who had, when you define extremely or extensively drug-resistant gonorrhea, it means it's resistance to ceftriaxone, which is our last fully effective antibiotic class for gonorrhea. So this is really very serious. I should also mention that at the IAS meeting in Munich, Jean-Michel Molina presented data from the ANRS DoxyPEP study for STIs in men who have sex with men, and they found a clone of gonorrhea with a signature plasmid that has the potential to confer high-level resistance to cefixime. So again, coming back to antibiotic use, widespread antibiotic use, we were talking about this again in the TV clinic this morning, lots of things we have to think about. The point of this slide, though, is to say that what we did here was to start the studies. We paid for and originated the early phase trials, so the preclinical work, phase 1, phase 2, and then we handed it off to something called GARD-P with another specialty therapeutics group for them to complete the international trial. And just was reported last year that this drug, zoliflodacin, totally new class, was safe and effective for uncomplicated urogenital gonorrhea. This is similar to something like the Medicines for Malaria project, where you really kind of get a consortium together and you say, look, this is too important to rely on industry. Sometimes industry partners will help, but I think we have to take this into our own hands as a global public health and medical community sometimes and make sure these things get done. I want to say a word about fungal infections, and I found this nice review of major drivers of fungal diseases in Africa. In the US, we have a lot of endemic mycosis. And living in the southern US, as I did for the last 7 years, I was amazed at how much invasive cryptococcosis in people without HIV we saw, as well as other invasive mycoses. But in Africa, this is a really, of course, big issue. And it's thought to be increasing in prevalence and also increasingly resistant to treatment. In 2021, there were an estimated 7,200 deaths from fungal infections. You may have heard of Candida auris. I don't know if you've seen any infections here, but that is a pathogen that does not have any antifungal therapeutics that really work very well. So we haven't seen a ton of it yet, but there have been outbreaks, particularly in hospitals, and it's very difficult to eradicate from the environment or if you're colonized with it. I've actually seen it in a transplant patient who probably had who had a lung transplant, and it was probably transplanted with the transplanted organ because it turns out, and we didn't look for it because we weren't routinely doing those cultures. So anyway, this person did fine, which is pretty amazing because it could have devastated the transplant, right? So it's really a problem. Lots of challenges to address fungal infections. I think it's a really important area, and I think we haven't again had a really big strategic plan to deal with this. It's not easy to find rapid and sensitive diagnostics. We have no approved vaccines, and there's a limited pipeline of new therapeutic agents. Of course, here I know you see a lot of cryptococcosis. There's been a lot of advances made in that. But that doesn't, I think, represent where we've gone and where we need to go in the future. Talking about TB here again is daunting, and I won't say very much because you could all teach me about TB much better than I can tell you. Just a reminder, in the U.S., it is definitely there and prevalent and probably underdiagnosed. We have about 13 million people living with a latent infection. Occasionally, we'll see outbreaks that get in the news. We just had several deaths in Southern California from a TB outbreak that was related to people living in close quarters in a shelter. Nothing like the burden that you see here, of course, and it remains the leading cause of death of people living with HIV and globally cause nearly twice as many deaths as HIV. I found this statistics shocking. I didn't really appreciate this, but coming back to my comments about antimicrobial resistance and how we cannot fail to include tuberculosis, drug-resistant TB accounts for about 1 in 3 deaths from antimicrobial resistance. So to separate out mycobacteria from everybody else makes absolutely no sense to me. So really important. We have a strategic plan for tuberculosis research that came out recently. I think you may have seen this given your vested interest in this. And these are goals that are really not going to be of any surprise. So in the interest of time, I won't belabor them, but I will say that we are supporting research in alignment with the WHO Global End TB strategy. And these are going to be familiar to many of you who do work in this area. So these are our key scientific TB programs and initiative. They're very, very active. There's FAST TB, which is trying to look at making sure everybody's on the same page, particularly in terms of who's studying what and how you are not duplicating, but you're complementing and you're really addressing the global portfolio. That sounds like obvious, but it's so complex when you think about how complex these studies are to do TB therapeutic trials. So I think it's a really important one. Novel diagnostics, of course, diagnosing TB, as we've talked about, is a huge challenge, particularly in pediatrics. And in HIV, right, where the disease often throws you a curveball instead of presenting classically. And then the tuberculosis research unit networks and the ones that I mentioned below. So the one that I also really just like to highlight is the TRACS, the TB Research Advancement Centers, because that is specifically tasked with promoting and developing young investigators. And I think TB is one of those fields where we have to make sure we have the next generation continuing to go in, because there's so much intellectual knowledge in people who've been doing TB for so long. The history of the drugs, the complexity of the regimens, the complexity of the way patients react to treatment, I think is just an unbelievable treasure trove, and it's important to really keep passing that on. Um, I know that you all participated, or many of you are part of the Imagine trial, which is looking at trying to treat TB meningitis. And I want to point out that one of the great things about this facility is your focus on pregnant women. If you look at many, many studies for everything in infectious diseases, often excluded pregnant and lactating women, even women who don't agree to take hormonal contraception are sometimes excluded. We're trying to change that, being very vocal about that. But one of the things that I really like about the work that you've done here is your pioneering work in that very vulnerable population. A couple of words about climate change. So, climate is a big issue for us in infectious diseases, as you know. And it's not just for the reasons that you think about. Most people think about this as a impacting vector biology. So you've got warmer waters, you've got warmer temperatures, the malaria, the mosquitoes, the sandflies, the midges, all of those little creatures are able to extend their range. And indeed, that is true. But you've also got more frequent disastrous climate events, right? So you've got people displaced, you've got disruption of sewage systems, lots more enteric infections. People displaced also often don't have access to their maintenance meds. So you have people coming off their HIV suppression. You've got then people trying to survive because they don't have food. They're selling sex for their drugs. So it is really a multi-dimensional, I think, catastrophe. It's really a tough one. And 2023, of course, was the world's hottest year by a large margin, if you were not aware of that. The other thing I should mention too is that livestock, right? A lot of people migrate, a lot of livestock migrate. So that's another thing, that axis of One Health, human livestock interaction, which I'm going to come back to in a minute when I talk about H5N1, is also an axis that can be really disrupted by this issue. The ClimateAid report is a great one. If you haven't seen the Climate Change and Epidemics 2023, takes you through all of this, gives you some really nice graphics and some interesting data. That brings me to malaria, which has continued to be a very serious public health threat. We've made some headway, but I think we are not where we need to be. I think we are poised in the next 5 years to make incredible strides against malaria. I actually think I feel that way about HIV and TB too. We can talk about that. But I think for the reasons I'm going to show you, malaria really represents an opportunity to have an incredible impact globally. So still a lot of disease. If you look at deaths by world region, of course, Africa is way in the lead there. And we're seeing a blip in 2020 that is going up. So we're seeing really a stall out of our progression in reducing morbidity and mortality. So the newly approved vaccines that we have, R21 and RTSS, are good. They're not great, right? So, and they're just rolling out in some countries. So hopefully they will start making a dent, but I don't think we're going to see that real fast. And I don't think they're going to be enough. So one thing that we've been very involved in and has come out of the Vaccine Research Center is the development of monoclonal antibodies. And this is a program led largely by Bob Cedar and others. But I just want to talk about it for a second, because it really gets back to this issue of investing in the basic science, the basic biology of understanding the immunology of these infections, and then essentially retro designing tools that reflect your optimal immune response. And that's what this group and many other groups worldwide are doing so beautifully now to generate medical countermeasures to the biggest threats. So you may recall in New England Journal 2022, there was a paper from this group looking at an antibody called CIS43LS, and this is a monoclonal that binds to the pre-hepatic phase of the malaria parasite. So essentially, you're getting infected by a mosquito, and you've got an antibody right there to bind to the circumsporozoite protein. And what this study showed that was that a single dose was safe and highly protected in Malian adults. And you can see that the time to the first P. falciparum infection over there on the right, you can see from 0 to 180 days, quite a large reduction relative to placebo. If you didn't have the antibody, about 80% of you were infected. If you had the antibody, that was cut down by 75 to 88%, depending on the dose. So that was incredibly encouraging. I have to say that, like, learning about malaria in medical school, seeing patients, I never ever would have thought a monoclonal antibody would have been a solution because it seemed too complicated. The pathogen just seemed too, evasive. But so this was really exciting, but it gets better. Another paper looked at a more potent antibody called L9LS, also binding that sporozoite, and published in the New England Journal in 2022 in a very small study, phase 1 study, that 15 of 17 volunteers were protected with controlled malaria. This is one of those controlled human infection models that are so fascinating, and that give us unbelievable data about how this stuff works. So it seemed to work really well. And beautifully, they went on to do this study, or to study this particular antibody in Malian children now 6 to 10 years of age, and just published earlier this year. Again, very similar efficacies to what we showed before, but in kids who have a much higher morbidity and mortality, of course, with malaria, particular when they get cerebral malaria. So it really worked very well in Mali. If you didn't get the antibody, you were infected 80% of the time. And if you did get it, depending on the dose, you were protected up to 70% of the time, depending on how you looked at the endpoint. So I think these are really encouraging. We are talking with many partners about how you would implement a larger study that perhaps combined some seasonal variation in when you give people this, how, when, what 6-month period. The other thing is that these seem to work for 6 months, which is pretty incredible. That's the other thing, that you could have a monoclonal whose durability is long enough to interrupt a season of transmission. So that's really, I think, visionary and something we would like to do, combined perhaps with some of these vaccinations, and of course with better bed nets, which bed nets, not bed nuts, bed nets, that we're getting. That may be another thing that we're getting. So I think it's very, very important. I want to say something about dengue. I know this has been a big focus of your surveillance, Tulio in particular. Dengue is almost out of control. Certainly in the Americas, it's invaded now many states in the United States. So now, of course, Americans think it's important, right? Despite the fact that it was killing people in many other parts of the world. It's dramatically increasing, and you can have incredibly severe outcomes, including deaths. So about 7,300 deaths in 2023. That is a big increase. And remember, of course, if you've had it once, you get it again, you're more likely to get dengue hemorrhagic fever due to the different serotypes. When you look at locally acquired cases reported, those are really recent data from the WHO. You can see that there are quite a few cases, particularly in Northern Africa, but I think a lot of this is just missurveillance. And that's, I know, something that you've been working on. So we do have a couple of vaccines, right, that are live attenuated dengue vaccines. People have been excited and interested in this. The challenge has been that the vaccines are not always active against all serotypes. And the concern has been, if you get vaccinated against one serotype and then you get infected with another serotype, is that like having a second dengue infection? And is it going to increase your risk of dengue hemorrhagic fever? Of course, in the Philippines, there was some suggestion that that was what was going on. But we do have now a Sanofi vaccine, a Takeda vaccine, and our vaccine called Butantan, which I think is super exciting. The two vaccines listed there are already licensed, but I'm going to show you some data from the Butantan phase 3 study. It's a single dose. And when you look at the constructs, you can see that it includes components of all four serotypes. So it's a little bit unusual and very exciting. This also was just published in the New England Journal, if you didn't see it this year. And this was one of the more exciting things I think that has come up since I've been on my watch here. This is a 2-year follow-up of the participants who got the vaccine. This is in Brazil, and you can see on the right-hand side, cumulative incidence of virologically confirmed dengue was markedly reduced by about 80% in the people who got the Butantan vaccine. Now, caveat here is that most of the disease in Brazil is serotype 1 or serotype 2. So we really can't say whether the components that were targeted to 3 and 4 are effective, but we do know that at least it looks safe, it looks effective, and we're going to continue to follow these people over 5 years. So very excited with this. I think it's going to be an interesting time. So in the last few minutes, I want to save some time for questions. I want to update you on what's going on with our highly pathogenic avian influenza outbreak. I know you have not, quote unquote, seen this here yet. We don't know. But this is a situation that is, I think, very, very concerning. And actually, another report came out last night that I didn't have time to add to the slides that I'll tell you about, which increases my level of concern. So what's the story with this? This is an H5N1 highly pathogenic avian influenza. You may recall that we have known about this since around 1996 when there was an outbreak in China on a duck farm. So that's an avian, it's an N3, H5, H3N7, I think it was. So there have been these circulating avian pathogens, flu viruses for a while. And we have seen cases, particularly in Southeast Asia, but we have not really seen an H5N1 2344B detected in the United States until March of this year. And it was very oddly detected in the milk of dairy cows. The dairy cows had mastitis. So they appeared to be quite ill. Their milk production went down dramatically, and that's what prompted people to look at that. We are now up to well over 20 or so states with infected herds in the United States. And these have been accompanied now by human infections in 13 dairy and poultry farms. And I'll talk about the human infections in a second. So far, we've been very fortunate because pasteurization works very well. It's a very bad time to drink raw milk in the United States. But of course, as soon as you tell people not to drink raw milk, what do? They go out and increase their consumption of raw milk because it's like a magical potion or something. And there's so many bad things you can get from raw milk besides brucellosis and the usual embovis and all that stuff. But now you really shouldn't be drinking it. Although GI acquisition and exposure with this pathogen is a whole other interesting question, and it's not clear what it does. Anyway, pasteurization works. The thing is that we are seeing this infection in multiple other mammalian species, and I'll talk about that in a second. Right now, if you look at that bottom graph here, that's the epidemic curve that we are seeing right now of the human cases. So back in March, that one case of conjunctivitis, 2 more cases of conjunctivitis associated with a dairy farm, but then 3 cases of conjunctivitis and respiratory infection in poultry farm workers who were culling infected poultry. They were wearing protective equipment, but the temperature was so hot. Again, global warming, right? It was like 105 degrees. They had huge fans blowing the air through so that people could actually tolerate being in the place where they were killing the chickens. And they couldn't keep their face masks on. So not only was there this incredible aerosolization, but people couldn't really manage to keep their protective equipment on. So it's a perfect storm. Many of these people too are undocumented people who are not U.S. citizens. They often go from farm to farm to try to get work. So it is a really, I would say, nerve-wracking situation. How do we know what's coming? I know you're very interested in viral surveillance. Were we surprised by this? we weren't totally surprised because we have seen this virus. We know where this virus is. This virus has been actually now in every single continent, including Antarctica, as recently as of last year. Huge marine mammal die-offs. So seals in the coast of Argentina, and in a lot of water mammals. So very concerning, dolphins, even whales. Why? Because birds. So I'm a bird watcher. I love birds. I hate to blame this on birds, but unfortunately, birds migrate, and birds, it's an avian influenza virus. So birds, many birds migrate from Southern America all the way up to the boreal forests of Canada or the northern hemisphere in a summer winter cycle. And so it's very probable that these birds were infected originally, probably around the sort of China outbreak or in that area. They then transmitted it north, have now transmitted it across the oceans. And so we have been able to track this. That's me actually on the right-hand side doing shorebird surveillance. So there's a number of groups. This is a group that we fund called the Centers for Excellence in Influenza Research and Response, and or the CERS, and they have many projects. This is on Delaware Bay, very close to Washington, D.C., which is a huge shorebird stopover. These birds come from, again, deep South America, all the way up to the Gulf of Mexico, stop in Delaware Bay, where something called horseshoe crabs lay incredibly rich eggs, and they gather there and they hang out there for, I don't know, 6 weeks, fatten up, and then go to the Arctic. So you can get in there in that window period and do cloacal sampling, which is what we do. You can also draw blood, which I think is really interesting, and people are looking at that. But these are enteric viruses in the birds. So that's me there. I'm not doing cloacal sampling, but I did sample many cloacas. That's a little ruddy turnstone there. Pretty amazing to think about these little tiny birds flying that far. About 24% of these birds are infected with this virus in their GI tract. And if you could figure out why they tolerate it and they don't get pulmonary infection, that would probably win you a Nobel Prize. So here's why I'm really worried about this, though. So it's not just the cows, and we're getting more information in the cows. There was a paper just the other day coming out from some of the CERS showing that the mastitis is a necrotizing mastitis. So these cows get very sick, and they probably are not going to be useful for more milk production, which is going to be a big hit to dairy farmers. Again, thinking about the potential for spread in a country like this, that could be really devastating. The other scary thing, though, is that we are seeing it in peri-domestic animals. So in this original report, there was a report of several cats. These were 2 cats who were found dead on a Texas farm. Now, the cats are farm cats, right? So they're running around drinking the milk. They're doing all kinds of stuff, and they're drinking pre-pasteurization milk, right? They're in the milking parlors, which is what they're called. So it's probably not surprising that they got infected, but the cats got really sick and died. And these are pictures of their brain, their lung, their heart. They had multisystemic necrotizing disease, very bad encephalitis, which, you can see flu encephalitis in kids. Those of you who are pediatricians in the room know this. It's really not that common in adults, and it's not that common in many mammals, but this was very worrisome. The thing that I'm concerned about is that just yesterday, Colorado Veterinary Medical Association reported 6 additional cat cases, 2 of whom were indoor cats and never went outside and had no links to any dairy farms or any poultry farms. So it's very clear that I think the virus is endemic. Honestly, right now, it's not going to be eradicated from the dairy industry. We know that the interaction between cows and pigs are the big species we worry about, right? Because pigs have the receptor for both avian and human influenza virus. So pigs are the ultimate mixing vessel for this virus. And when it gets into pigs, that's going to be quite alarming. So we're doing surveillance. USDA is doing surveillance. But I do think that with more cases occurring in humans, with now a finding in true pet cats, that could be very, very worrisome. So just a couple of slides to show you some of the work that our groups have done looking at the fact that pasteurization does work, but it's also not easy to clean up the places where milking is done. The virus is contaminating everything, contaminating the surfaces. These are not sterile spaces, right? Milk is not sterile. So you can imagine that when you get in there, What the workers do with that machine that I had in the first slide is just to wipe it off with a wet rag. So, sterilizing that is really kind of out of the question. So how do you do that? How do you protect workers? People rub their eyes. They're not going to wear protective equipment. So it's a kind of I think it's a mess, to be honest. But so just so you know what's happening there, and if we have time, we can talk about countermeasures, but I don't want to go over time. So I'll just mention this is, I think, a nice slide of what we do in terms of our investigational product activities. Again, many of you know that we have worked with you, and particularly the Afrigen folks, trying to look at mRNA antigens. But these are some of the things that we're working on in the Vaccine Research Center. And I bring this up because we are actively working on this in terms of H5N1, particularly for monoclonals, because my fear is that if we start seeing cases really, really explode, I don't want to be alarmist, but I think we need to be prepared. We do have a vaccine stockpiled. It probably is going to help, but I think monoclonals may be necessary to get people over that. I know I'm almost finished. Let me just show a couple of slides on Mpox, because clearly you all are seeing quite a bit of it. I will not go over the 2022 outbreak, but I just want to point out we are painfully aware of the fact that the Central Africa situation with Mpox is rapidly changing. We're seeing more disease. We're seeing fatal disease in kids. And we really don't have great treatments for this. So there was a record number of Mpox cases in the DRC, and we're now seeing it in neighboring countries. And this is almost certainly both transmitted through direct skin contact, particularly in kids, but also sexually transmitted. So a really big problem, and this is just a little bit of the lineage. And then spreading in Cape Town, you have had 20 confirmed cases with a death toll of 3. So just a summary of our Mpox research. We are sponsoring a couple of studies of tecovirimat, particularly STOMP, a number of ways to try to look at extending the use and reservoir of the vaccines we have, but also looking for better diagnostics and antivirals. The last thing I'll say is just that I really, as I know many of you are concerned about the pipeline of young investigators, and not just physicians, but all the people who work in our teams, the nurses, the biostatisticians, the data entry people, the quality assurance people, the pharmacists, everybody, the counselors, because if we don't invest in our teams to continue to do the work that you're doing, we can have all the cool tools in the world, right? We can have these great tools, but we can't study them, and ultimately we can't we can't implement them. So I just want to point out that I'm going to cut through this really quickly, that we just can't forget that people are the unifying element for doing what we do. And if we don't have this pipeline of researchers, and by researchers, I do mean the whole team, then we're not going to get where we need to go. So I'll stop there. Thank you for your patience again, and I really appreciate the opportunity to talk with you today. Thanks.
Enjoyable. Let's give a few moments for questions. Yes, please go ahead.
Mariah? Yeah. Another thing, just to warn you, we ask lots of questions. Yeah, and that's very characteristics of this seminar. Yeah.
Yeah, thank you for your talk. I was wondering about the influenza outbreak you were telling about. A few weeks ago, we had a training session here with a lot of African fellows and studying this particular outbreak. And one of the things
H5N1.
Yes.
Sorry, I couldn't hear.
The H5N1, yes. And one of the things the fellows mentioned was they are also doing sometimes research with farms where it's actually quite difficult to access the farms because they're not so open to study, for example, antimicrobial resistance. I was wondering what your experience is in that, and maybe also in more general, what are your lessons from this outbreak and how the US responded to it, and what we can learn as well from that?
Terrific question. I would say, having been on lots of calls with lots of the other agencies, many of the agencies have worked together. So when you think about this response, It's not just CDC, it's FDA, it's NIH, and critically, it's U.S. Department of Agriculture, right? And it's also state health departments. So when you look at state health departments, some of them are staffed up beautifully, like Colorado and Michigan are fantastic. That's where the cases have been reported from, which makes me believe that there are many other cases in places that don't have the infrastructure to really do the kinds of investigation we need to understand what's going on. So that's the first lesson, that you have to have a good relationship with and trust and well-staffed infrastructure to try to figure out what the epidemiology actually is. The other issue is getting on the farm. CDC investigators have not been able to get on a single farm until last month, when they actually got onto the farm with those poultry workers who got infected. And that's because the mistrust in government, the desire to continue to have a business when you're trying to support, your family and your business, they're really concerned that if word gets out that this is the farm that had the infected cows, that they're going to get shut down. So I would say the combination of mistrust of government and regulators, and also our fragile public health infrastructure, are two things that, if anything's going to take us out in this or really, really hurt us, it's going to be those two things. Great question. There's a lot more to talk about there.
Great talk. Thank you. The Wellcome Trust, Max Planck Institute, the British MRC, they all have international centers with established African, for instance, or Southeast Asian institutions. Does the NIH in any way consider doing something similar? Because there are several universities in South Africa, we and UCT and UKZN and WITS, who have long-lasting collaborations, close collaboration with intramural researchers. Is the NIH in any way looking at establishing similar centers in Africa?
Are you talking specifically about pandemic preparedness or?
Generally, I mean, there's all the topics you really touched on, pandemic preparedness, TB, HIV.
Yes.
They would all.
Yeah. So I think it depends on what you call a center. Given all of the research that we fund, there's actually, I think it's in several buckets, right? So there are collaborative networks. Certainly the HIV, the TB networks are already well-established collaborative networks. Some of the CREEDs, the Centers for Emerging Infectious Diseases, are very much reliant on centers in Africa. So I would say, even if they're not called a center as such, you've got really strong relationships that you mentioned. I mean, I think it's worth talking about whether there would be a benefit to thinking about it differently, sending a different signal. The other thing is that we want to make sure that people have access to independently funded and proposed research. So a lot of you today told me about your R01s, which is music to my ears, especially after coming off Fogarty's. So to me, yes, the centers are great, but I always want to keep in mind that center grants tend to favor senior established investigators, which is great. They do great work, but we also need to make sure we have resources to support emerging investigators who are well mentored, which is not always the case, frankly. So I think fantastic, let's talk more. But I think that we are really committed to working with many places in Africa on those issues.
Thanks very much for a fantastic overview of your of the NIH's infectious disease spectrum. One, I suppose, question/comment. One's a very specific one related to the focus on antimicrobial resistance, which I was very pleased to see, and I must say I wasn't that aware of. So very good.
That's great. I'm glad I came.
Is there any discussion around kind of funding research programs around infection prevention and control, which is probably one of the biggest, or I think, most cost-effective ways of managing AMR?
Yep.
And then the other question is a more transversal one. When I think very often TB researchers and HIV researchers and AMR researchers work in their own silos and don't always communicate.
Yes.
And the seminar is a great way of trying to break that down. Is there any strategy within the NIH of how what's going on in the TB world can inform the AMR world looking at diagnostics, vaccine surveillance, and so on.
A great question. So with regard to your first question, this is something we have not. So when you look at hospital epidemiology, infection control, we do not have a big footprint there. And that's been noticed by the people who do that work, which is a very large contingent, right? And it's not just MDs, lots of infection control practitioners. I mean, it's a huge thing. And it became a huge thing, of course, during COVID. People really, I think the spotlight was there. The challenge really is, what should the research strategy be? Because it's difficult, expensive, and very time consuming to do randomized controlled trials, as I know you know. And I'm not sure that's what we need to do here. I feel, and I'm not an expert in this area, this is just my take from talking to many people who do this. I personally think we need to have some sort of consultation about the strategy here and how we can really advance that field, because I don't think we have done that. It's been a blind spot for us. I think in part because, again, it's often not basic science based, right? We haven't funded that area. But I think it's time to really talk about it. Your second question is really near and dear to my heart, because I have been talking a lot about how we are a very classic pathogen-focused institute, right? We love our infections. We love our pathogens. We are TB people. We are hantavirus people. We are malaria people. But patients and people in the community that's not how they define themselves. And they often have more than one thing. So how do you think about a whole person approach that is agnostic to what institute the person happens to be, studied in? I think we have to move towards that. I don't know, you can't start by blowing up the networks that are devoted to pathogens, right? God forbid. That's not what I'm proposing. But we are getting better at, I think, for example, the AIDS clinical trials units groups is now doing TB vaccine research and people living with HIV, right? You had them involved in the Reprieve trial, looking at statins in people living with HIV with normal cholesterol. So TB and HIV, I think, are logical. We've had more success in getting funding that's been allocated to HIV to study TB in people with or at risk for HIV, to study hepatitis C and B, which I think is huge. Still way too much hepatitis B in Africa in particular. I was looking at that the other day. And then also STIs. So for example, we have been able to use some of our HIV resources to further the study of the meningococcal vaccine for prevention of gonorrhea, which I would love to talk about. That was my study before I came to NIH. So we're doing that now in Malawi. So I think we're getting there, but that's a big shift. And we've got to talk our friends through it, because I think some people are very threatened by it. I mean, Steve, you've been in HIV for a million years, but you've been doing a lot of stuff. So I don't know if you want to comment, but yeah. I think can we give him center? Yeah, the center. Please comment on that.
I just want to follow up about the question about the NIH center. So I am an intramural clinical scientist, and the intramural program back in the late 90s developed an idea called the International Centers for Excellence in Research. So the first one was in Mali, which focuses on malaria vaccines. And then I run the one in Uganda, which is predominantly focused on HIV, but has branched out and is doing a huge range of things. And the idea is exactly as you envision, that we would have these centers that would support collaborations with African scientists and really build capacity through training and research. So the Institute does envision that. They're expensive and they require a long-term commitment. So they, we don't have the resources, unfortunately, to have one of these everywhere. But there is a commitment from the Institute to build capacity through those longitudinal relationships, which complement the extramural world as well, because the center that I work with collaborates with an entity called Rakai Health Sciences Program, which has a lot of extramural funding as well with our colleagues at Johns Hopkins. So there is a commitment. It's a great thing. We'd love to do more, but we need bigger budgets to do more.
Yeah, thanks, Steve. And I should have mentioned the ICERs specifically, the International Centers for Excellence. That ICER in Mali is one of the reasons that the malaria vaccines, malaria monoclonal antibody studies got done. We have an ICER in Cambodia where we're actually getting PBMCs from people who have been infected with H5N1. So we can really look in real time again, interrogate those immune responses and hopefully use those. So thanks, Steve, for fleshing that out.
Yeah. I have a couple of questions. Yeah. And then we have 100 people online. We ask if they have a question. Yeah. So one question, given that the NIH changed director, and do you foresee the research and funding going for HIV to decrease in the next few years, or do you see it keeping stable?
I don't see it going to decrease, although I think we can't be complacent because you saw what happened with PEPFAR and the threat to PEPFAR funding. So, and that's only been assured for a year. So I think the more we can do to talk about how we use money specifically for HIV to improve the quality of life for people, regardless of what's going on, whether it's TB, again, hepatitis, whether it's reproductive health care in people living with HIV. I think anything that we do is going to be hopefully good justification for continued funding. We have not heard, I would say, any clamor to decrease the funding. And I would say there's a very active advocacy sort of coalition to hopefully keep that going forward.
Okay. And can I ask you 2 difficult questions, or should I just be polite?
I'm not really a fan of polite.
Okay, good.
Unless, we're at dinner.
Okay, because I'm trying to be my best behavior, but doesn't last long. Yeah. So one question, yeah. So we understand that South Africa is the biggest funder country outside the US, and we also understand the impact of the research. Yeah. But is there any chance to change the way that things are funded? So, for example, at the moment, to hold the NIH grants for us, it costs us money. Yeah. We receive 8%, less than 10% overheads, where the John Hopkins in the Haikai, I think that's 75. Yeah. Yeah, higher than that. Is that any way that could be discussed further? Because especially if it is one of the vision, we want to invest in the younger generation of scientists, but at the moment, to hold a grant, it costs us money. Yeah. So any foresee change on that?
I think that's a really good question, and I've heard it repeatedly on this trip. So it's clearly a pain point. These are policies that are, I think, and Sophia can tell me that I think these are HHS policies. And they are certainly NIH wide policies. I think it's a fair question, and I don't know when that was established. I think it's been that way for many years. So I think it's really reasonable to have that conversation, and I'm certainly will bring it back and ask. I really don't know what the likelihood is that they're going to change. Our budget. Even though we sound very rich, our budget is very tight this year. We had to make some really painful decisions about our pay lines, and I'm sure we're going to be, our fiscal year ends September 30th. I'm sure we'll be on a continuing resolution given the election. So even though it sounds like we have a lot of money to throw around, I wish we had more, and I think we need more. So it's a really good conversation to have. We really needed to hear your experience, the effect it has on you, what you would do differently if you got more. And I think we can bring that back and talk about it. I don't know, Sophia, do you want to say anything? Or is it like, yeah.
And
And
it's part of
Sophia,
our
introduce
regulations.
yourself.
It's not an
Introduce yourself.
NIH thing. And Joelle can comment on that as well. And this has come up before. I mean, it's basically a obligation of how U.S. taxpayer dollars are spent, both domestically and externally, and how we maintain the scientific ecosystem. So the differences that you're talking about come from that. But on the flip side of this, South Africa actually, and in your government, invests money in research as well. And we really are grateful that they are doing that. And we hope that they will continue to increase and fund and build that ecosystem here as well, because you are so critical to responses we have seen more recently in the pandemic.
my last question. Yeah, because I think that you experiment that in person now with the H5N1. Yeah, we have this great interest on funding research on virus discovery, zoonotic transmission. Yeah, but we have no incentive for people to do that. So we were an example. Yeah, that by discovering a variant, we got punished with travel bans and discrimination, and now you have the same, unfortunately, with the agriculture and the vet people, that they prefer not to describe to do that. Yeah. So at the moment, I'm in big discussion on the pandemic accord by a very high level at the WHO. It's any way that the NIH can think about the shared benefits of such important research, especially as zoonotic transmission and new emergence is quite high in your agenda.
Yeah. So this is a great example, and there are other disincentives, right? Because you have probably seen many of our scientists who do virus surveillance have been vilified, for studying bats or for studying you name it, right? Because they're going out there to try to find the really dangerous viruses. I mean, there's a lot of misinformation out there. So that's a big disincentive, because you don't want to have to, be concerned about your reputation, even if scientists know what you're doing. I think the challenge is that, let's go back to the variant discovery. We know what needed to happen. We know what shouldn't have happened, which was the travel ban. But the people who decide those things are often very divorced and just not really. I mean, we only have so much impact as scientists. How can we create, I think, a better communication, a better interagency agreement? I'd like to think we got to learn more from the pandemic. I think there is a path for better communication around that. If the same thing happened today, would we be able to reverse that kind of decision? I don't know. I'm not sure. But I agree with you. It's a huge problem, because you're punishing people for doing the work that they should be doing. And I don't know how we change that. I can give you an example with H5N1. CDC and USDA are starting to financially compensate people. So they actually are giving farms money for the cattle that are lost to disease. And I think that if you're going to start to get people to disclose and share information, you have to recognize that this could impact their livelihood, and you have to make sure there's a safety net there. So you need to allocate money. I mean, it's really about making sure people feel comfortable telling you what they need to tell you. So how you do that, I think we're getting a little bit better, proactively thinking about it, at least with the dairy farm thing. I think it's getting a little bit better. But that has to be part of the conversation early on. So I'm sorry that happened. I think it was very unfair.
It's no problem. We can send you our account number from our government. Yeah, we estimate we had a piece in the Lancet was something as like $6 billion, a billion rand that we lost from the US. It's not for me. I can send the treasure number, but thank you so much. Yeah, for being
thank
here.
You for all the work you do. I really appreciate it.
And thanks so much for accepting to do a seminar in your busy schedule. And now we're going to break. We have some drinks and food being served here, so we can engage with our great visitors from the NIH, from the US Embassy. And we really appreciate your support. Yeah, I think that South Africa and the NIH work really well. Yeah, and I think that together, I think that we can really help